Tuesday, we talked about amino acids, right? What we're going to talk about today is how these amino acids come together to form peptides and proteins. And then we'll talk a little bit about protein structure. It's primary, secondary, tertiary, quaternary. You may have learned that at some point along your journey. And then talk a little bit about some consequences of protein misfolding. But I put this link here. There's this article that just came out recently where they are taking protein folding.
Oh.
Did you see this?
Yes, I saw it.
Did you listen to it? Yeah.
So cool.
Yeah, and they turned it into sound. So for some reason, I don't know how to connect to sound. I don't know if sound is even in this room, which
I think Kathy had problems with it too.
yeah. So I don't know. I'm gonna try to play it on my phone and on here at the same time and see if we can hear it. May not be synced. Oh, that's pretty good. So it's almost kind of like a roller coaster ride, you know, but it has these clicks that associate with different turns, different angles, and each of those turns and angles are dictated by the amino acids and those properties on those side chains. And so predicting protein folding is a challenging endeavor, to say the least. With AI, it's making it way easier. There's a game that came out years ago called Foldit, which was a citizen science platform where non-science backgrounds can put together the pieces based on hydrophobicity, hydrophilicity, hydrogen bonding, sulfur bridges, all of these components, and predict protein structures and then see how close that is to the actual conformation. And so between that and AI, and then now applying clicks and sound, they did that too with, I remember during COVID, for the COVID, so for SARS-CoV-2, the virus that causes COVID, the genome they sequenced and they applied computationally notes to each type of nucleotide, or maybe like trends of nucleotides, and they played it. It was a symphony, if you will. It was not just clicks, it was music, and it was a little bit disjointed and strange, but there's some creativity to that. Science and art, melting together. Okay, so what we'll do today is I want to go over those practice problems that we skipped over. Maybe you had a chance to go over them between Tuesday and today. If not, we'll do it today. And then we'll talk about peptide bonds, how peptides are synthesized, get into structure, denaturization of proteins, and then proteins have a very important function that is in the form of enzymes. We'll talk just a little bit about enzymes. All right, any questions or comments or concerns before we get started though? No, anybody heard from Nikki?
No?
No? Okay.
I saw her yesterday, but haven't heard from her today.
All right. So we kind of ended Tuesday talking about how to synthesize amino acids. And the amidomalonate was the first one we discussed. And then we also talked about Strecker synthesis, where we use ammonium chloride and sodium cyanide. And then we use a really strong acid to essentially create whatever amino acid we're looking for. It's going to require a certain starting material, right? So if we wanted to make alanine, we would start with essentially the basics that has that R group attached to it. In the case of alanine, it's just going to be on that alpha carbon, a methyl group. Okay? So we start with a product with a carbonyl and has our, the R group is going to be directly connected to the carbonyl. But then once we go through this process, we end up attaching that R group to the alpha carbon, which is the carbon connected to that carbonyl. Okay? So it's not going to be directly, it's no longer directly attached to this, because then it wouldn't be an amino acid anymore. You know? It's going to be attached to that alpha carbon. Okay? All right. So for this problem, we got to look up, do you guys have your structural sheet for these different amino acids? Okay, so let's start with methionine. Is that the first one?
Mm-hmm.
Yeah, okay. So methionine is going to be what? Well, thio and meth kind of gives it away a little bit. We can draw our general structure of an amino acid. Okay. So, what else do we have for methionine? What's the R group?
The R group. It's like, I'm trying to remember.
It's 2 carbons,
Yeah.
then a sulfur, and then a methyl group.
Yeah.
So that's what we're trying to get to.
Mm-hmm.
Question is, when we use ammonium chloride, sodium cyanide, and H3O, what is our precursor? It's just going to be that group attached to a carboxylic acid. So, is that right? 1, 2. 2. Oh, need one more. Okay.
Isn't this one? Is this the one that it adds a carbon with the nitro in the synthesis? Or?
Yes.
Okay.
That's why we're getting, right? We have 1, 2, 1, 2. So if we look at the carbons here, this is carbon. This is going to be the alpha carbon. This is the beta carbon. Is that right? So then, for histidine, what's that going to look like? Histidine, I think, is just a hydrogen? No.
Oh, it's
What's histidine? That's got a ring?
a ring with like nitrogen inside of it,
Okay.
like 2 of them.
This is why I don't make you memorize these, because I memorized them 20 years ago and I don't remember all of them. So, coming off our alpha carbon, what do we have for histidine?
From the alpha carbon, so there's like 1 carbon in the middle, and then it's cyclopentene, but it has like a carbon at like 2 and 4, or a nitrogen.
Usually
Yeah. And there were like double bonds from the nitrogen on the left going down and then like adjacent to it,
That?
yeah.
No.
It's like from the first carbon going up and then from the nitrogen going down. That's how it has it in the picture.
Okay. Look good?
Mm-hmm.
All right. So, to get there, I'll let you fill in the missing pieces there.
Question for the streaker synthesis. Last week it was shown to like start with an aldehyde. Can you use an aldehyde and a carboxylic acid, or does it have to be one or the other?
You can use an aldehyde too.
Okay.
Yeah.
So either one.
In fact, I think we do use an aldehyde. Yeah. Sorry about that. We have to use an aldehyde.
Okay.
Yeah. That was my getting lost in translation of that. So not a carboxylic acid. We're just going to use an aldehyde here. Okay. Thanks for catching that, Bob.
Yeah, I just wanted to clarify.
All right, so we just need to come off here and then have our ring.
Mm-hmm.
Okay, cool. What else? Phenylalanine. So alanine with a phenyl group, right? So it's going to be alanine. So coming off that alpha carbon, we have an alanine and then a phenyl attached to that. Here, so. Here. Does that look right? Yeah? Okay. So, I'll let you come up with what we need for that. And actually, so quickly, is methionine, is that correct? Because there should be, there should be one more carbon, either one less carbon here, or one more carbon here.
Yeah, I was thinking maybe one starting with one less, because then that nitrile comes in. I was looking at the mechanism,
Yeah.
and it looks like that.
Wait, so
Nitro
the
comes in
nitrile
there.
adds the carbon?
Yeah. How I was looking at is like the carbonyl kind of splits into like the amine and then the carboxylic acid.
Mhm.
We don't have enough carbons on that methionine.
Oh, okay.
So there's the alpha carbon.
Uh-huh.
So 1, 2, 3, and
Then
then
sulfur.
S.
Oh, okay.
So this
That one's right.
is right. Here, 1, 2, 3, then S. There we go. Okay. And then this one. Our aldehyde, and then coming off there, how many are we adding? 1, right? So then there's another carbon here.
Mm-hmm.
So there's essentially just, there's the 1 carbon and then the ring coming off,
Mm-hmm.
right? Okay.
Yeah, that's what I had.
All right. And then leucine. Is that leucine?
Yes.
Okay. So what's the precursor for that? You know, essentially what you could do is you could just take off that amine group, right? And then move this to the carbon up here. So and then all right, cool. So that's going that direction. This is going the reverse direction. I'll let you work on these on your own if you didn't already. It's giving you these products, and you can see that, yes, Rob, they're all aldehydes.
Okay.
Not benzo or not carboxylic acid. So chugging along, though, let's talk about peptide synthesis. So a peptide is literally just 2 amino acid residues, and the residue is what we call the different amino acids. Okay. Residues are subunits that come together to form a peptide, which is going to be 2 or more amino acids. Okay. And so the one they're showing here, this is glutathione. You'll notice that it's not very big, it's just 3 peptides. Glutathione, glue for glutamate, and thio for the cysteine that has that sulfur group. And glutathione is an important peptide for essentially scavenging around the body for free radicals and electrophiles to help prevent those things from attacking our biomolecules, because as you probably can imagine, this far into organic chemistry, if you change a molecule in a very simple way, right? Add on an oxygen, take off an oxygen, you radically change that molecule. They can look to the untrained eye identical and have completely different properties, right? So we have certain things in place, antioxidants and stuff, to help sop up like a sponge those free radicals and electrophiles. And just like changing those biomolecules have different, vastly different repercussions, the order of glutathione has to be this glutamate, this cysteine, and this glycine residue. If we were to switch them around in some way, it would no longer confer those properties. Okay, so even just switching around 3 amino acids can radically change what that peptide is going to do and can do. Even if we just have 2 amino acid residues come together, that is considered a peptide. Okay? And if we have alanine in front and glycine in the back, then we get this essential, isomer. We can switch those around and get a different isomer. These are constitutional isomers, but they can have different properties. And we'll talk about, we think about this as a directional molecule. So to the left is going to be the N-terminus, and it's the next slide or the next couple slides. And then the right side is the C-terminus, N for nitrogen, C for carboxylic acid. So we're always going to orient peptides when we're analyzing them from left to right, and that being N to C. All right. So here, alanine is the N-terminal residue. Glycine is the C-terminal residue. And if we switch those around, it can have different properties, because now here the alanine has that amine group on the outside, but here the glycine has it. Okay? So alters their behavior, alters what they can bind to, what they can't bind to. So yeah, here is that. It's drawn here like a puzzle, right? Because we often think of these amino acids coming together in puzzle piece-like formations. Essentially, the N-terminus, like I said, is for the amine group, C-terminus for the carboxylic acid group, and what's going to happen for this synthesis to occur, for these things to come together, is the OH group on that carboxylic acid is going to pop off. And then the amine group is going to lose a hydrogen. Okay, so that's going to be a dehydration reaction. So you're going to lose a water molecule, and you're going to then stitch those together on that carboxylic acid carbonyl. Okay. And again, there's a directionality to this. Okay. Because you can imagine that if it were backwards, then you'll get kind of a reverse folded protein. Right? If you took a sequence of 30 amino acid residues and just reverse them, once we talk about the folding patterns and stuff, it might make more sense, but hopefully maybe you can just kind of imagine in your mind that reversing the sequence will give us a reverse folded protein, and when it comes to protein conformation or protein shape, that's what dictates the function. So if we reverse this, we have a totally different shape. It's not the same protein at all.
Because it starts folding as it's being made, then, or?
So usually, it can, and we'll talk about that a little bit, but the mRNA, if we're making this in a biological system, right, not just in the lab, the mRNA goes to a ribosome. And then the ribosome is a protein. It chugs along and it reads 3 nucleotides at a time. That's called a codon. And every 3 corresponds to a specific amino acid. So the transfer RNA brings in that amino acid, right? And then it keeps reading, and then they're doing this process here, right? As that ribosome is coming on, there are enzymes, other proteins, that are facilitating the next amino acid to come on and bind to that. And then it reads another 3 codons. Transfer RNA brings in the corresponding amino acid. And so you get what's called the primary structure first. Okay? It might be doing some folding while it's still attached to the ribosome, but a lot of that folding is going to happen once it's released from the ribosome and then enters like the Golgi apparatus. Because at that point, they may need to add sugars to it. There'll be what's called post-translational modification. So after that protein's translated, it can still be modified in certain ways, glycosylated or sugared, and then that says, okay, you're going to go to the cell membrane. You're going to stay in the cytoplasm. But if that sequence was backwards, now what's going to be an N-terminal portion of the amino acid would be flipped. And so that R group is also flipped. And so it just gives you a completely different shape.
That's interesting.
So glycine, alanine, this is what it looks like in the bond line drawing. Oftentimes they just draw them as circles, and you can see here the consistency of these amino acids. And again, alanine, glycine, cysteine, tryptophan, glutamine, phenylalanine, cysteine, methionine, etc. If we reverse this, then again, it's not going to be the same. And the reason it's not going to be the same is because all of these residues, if you were to look at the chiral carbon, they would be an S configuration. One exception, that being cysteine, that sulfur group. Everything else is S. So with that configuration, if we're drawing them out like this, and we have our N-terminus and we have our C-terminus, the way the R groups are arranged is that they alternate. Down, up, down, up, down, up. And in doing so, right, these carbonyl carbons also alternate. And since they alternate, and since they all, except for cysteine, have an S configuration, the R groups on the top can all be wedges. And the R groups, so so I should have brought some props, right? But, you know, like we have this chain of a bunch of amino acids. All of the top R groups are coming out toward you, right? And then all of the bottom R groups are going into the board, which is convenient. That makes it nice. But again, if we were to switch these around, then we switch the R groups. Okay. So it's practice. Let's draw. Essentially, let's draw this with however many amino acids, and let's have wedges and dashes for the R groups. What do they want? They want phenylalanine, valine, and tryptophan. So I'll give you a few minutes. Look at your cipher and draw those out. See what you come up with.
The previous slide, can we get a copy of that? It somehow
Doesn't get in there yet.
Yeah, I added that at the last. Yes. I will do my best to remember to add that. Still working?
I think I got it.
Yeah.
Got it? Okay. Phenylalanine. So, again, you can make it, doesn't really matter. You can make it wedge or dash in this case. If that's a dash, this would be a wedge. And, ooh, valine. What's valine?
It's like an isopropyl.
Isopropyl. So that.
I think so, yeah.
Okay. And then tryptophan. And dash. And then.
It's like it's nitro cyclohexene.
Okay. Well, you've written it down, so I'm gonna be lazy and not write it down myself. But either wedge dash wedge or dash wedge dash. Just make sure you don't have too many carbons sandwiched in there. But that's essentially how we do it.
So I ended up drawing the first one like going out the R group going up the top with a dash. That's still correct. If
Yeah.
it's like just a different way of looking at
Yep.
it. Okay.
Yep. That's fine. Yep. Absolutely.
Okay.
So your carbonyl is pointing down, but yeah, it doesn't really matter in that case. All it matters is that the carbonyls and the R groups are alternating,
Okay.
right? And then everything on top, all the R groups on top have the same directionality to them. All right, questions in addition to that one? All right. What's next? Disulfide bridges. So, anything that has a cysteine is going to have a sulfur, right? And if there are two sulfurs, they are going to form a disulfide bridge. And this is going to be really important for 3D structure of the protein. Hydrophobicity and hydrophilicity guide the shape of a protein. Hydrogen bonding guides the shape of the protein. Disulfide bridges guides the shape of the protein. And it can guide the shape of one peptide, right? Or it can link two different peptides together. And jumping a little ahead, when we get to quaternary protein structure is when we have more than one protein complex together. So insulin is an example of this. Insulin are two peptides that are linked together by disulfide bridges. And it can get really, really complex. So DNA polymerase, stuff that you used in your PCR reactions, I think those have upwards of 10 different proteins all complex together, hooked together, joined by these sulfide bridges. Okay. It's also what makes hair curly or straight. Your hair is keratin, which is a protein, and the sequence for your hair, if it has a lot of cysteines in it, that's one of the things that can give it its curl, right? And if some hair's straight, some hair's wavy, some hair's really curly, and that's guided by, in large part, some of these cysteines. And so if you've ever had your hair chemically straightened, which I haven't because I don't have curly hair, and if I did, I don't think I would mind it, but the way that's done is it destroys the disulfide bridges, and so the hair as that helical shape relaxes.
Isn't that the chemical that they bring up in Legally Blonde in the trial?
I don't know. I don't think I've seen that movie. But it could be. Could be.
And there's relevance in chemistry.
I will have to check that out. That's with. Don't tell me. Well, shoot. Now her name is escaping me. But she was. What's that?
I can picture the actor, but I can't think of her name either.
You know.
Reese Witherspoon.
Yes, that's it. Reese. Yes. Okay. I'll have to do some homework and watch that. So here are some interesting peptides that are in some cases different only in the C-terminal amino acid residue. So in enkephalin, we have leucine enkephalin and methionine enkephalin, and it's just a 1, 2, 3. It's a it'd be a pentapeptide, right, or a pentapeptide. Even though these are the same number of amino acid residues, they have this difference in this last residue, leucine versus methionine, and that said, they're both linked in controlling pain. So it's believed that these two are bound to the same receptors that morphine binds to. So just these really short peptides can be linked to that physiological response to pain management. So I think in terms of getting desensitized to certain pain, right? Eventually, this would build up and bind those pain receptors and block them. Okay. And then bradykinin, what is that? That is a hormone. It's an anti-inflammatory hormone, I believe, and it also is involved in vasodilation. I'm pretty sure. I could be wrong. It's not going to be on a test, so you can back me up if I'm incorrect. I believe bradykinin is a vasodilator, and also because of that allows the influx of anti-inflammatory peptides, including itself, to tell the immune system to calm down. Right? So you may know that being in a chronic state of inflammation is not good. Inflammation is our first line of defense against pro-inflammatory agents like microbes or other non-self entities that get in the body. And so the inflammation is just all of these cytokines, which are also peptides. And they're a messenger signaling system to tell your immune cells to come in and start attacking and cleaning up whatever that foreign invader is. But sometimes your immune system gets so pumped up that they need signals to say, hey, I call it the Taylor Swift. You need to calm down. You know her song, Calm Down? You need to calm down. All right, and so that's, I think bradykinin is one of those that does that. Vasopressin, oxytocin, you might have heard of these. These are interesting in that the C-terminus is modified. So the C-terminus, instead of having the OH group, it has another amine group in place of it on both this and this. And these are examples of peptides that are the same in number, both have this modified C-terminus, and differ just in the position of the third and the 8th amino acid. And because of that, they have pretty different functions, even though they have a very similar structure. So, oxytocin, you may have heard of that in terms of the cuddle molecule. Some people have little jewelry that has the oxytocin molecule on it, and it's thought to be released during intimacy and stuff. It makes you feel more sociable around people. It's more, I don't know, scientifically validated. Use is in inducing labor. So it's a natural peptide that's released during induction of labor for pregnant women, and then promotes milk stimulation. So lactation, I believe, doesn't occur till after, toward the end of the pregnancy, or really ramps up after the baby's born. And that is signaled in part by a profound release of oxytocin. Whereas vasopressin gets its name as, again, a vasodilator. So it's going to regulate blood pressure. So very different from inducing labor, regulating blood pressure. Both of these have actually been, I was listening to a podcast not long ago on autism, because my third son is mildly autistic, and so they were talking about oxytocin and vasopressin because these have been found to be under-regulated in people with autism. So, and it was interesting because this woman was talking about this. Before she was a scientist at Stanford, studying, I believe she researches in neurobiology. She was looking at the social behavior of, I think, groundhogs or prairie dogs, okay? And found that males that have less vasopressin in particular are less sociable. And then when their partner, I believe they mate for life, when their partner has babies, vasopressin gets upregulated and they get a little bit more pleasant to be around. Okay? And so there's this link potentially, in addition to vasodilation, blood pressure modulation, labor induction, milk production, there might be this cuddle molecule aspect, which also could be associated with why some people on the autism spectrum have challenges with being social, essentially. I think that's a lot of hand waving right now at this point, but the illustration I'm trying to make is that this peptide can be tweaked slightly and have very different functions, and can have multiple functions. Like it will never, I hope this never causes me to start lactating, right? But I have oxytocin, but it could be involved in the degree of my social nature. So that's fascinating to me. All right, let's take a break. Let's take an 11-minute break and come back at 12:05, and then we'll just, because this is OCHEM, we got to do some mechanisms, right? We'll go into the mechanism of how these peptide bonds are created, and then from there get into a little bit of protein structure and a bit more relaxed stuff.